Living things are complicated, as you can see in this picture here, and biophysicists are trying to figure out how life works. Hello, my name is Oliver Beckstein, and I'm an associate professor in the Department of Physics here at Arizona State University. In my lab, we are trying to understand how life works. Living things are made from cells. The cell is the basic unit of life. But cells themselves consist of molecules, and molecules consist of atoms. So the idea is, if we understand how the atoms interact and form molecules and how the molecules interact, then we can get a bottom-up understanding of the processes that make up a living thing. Because the molecules that we are talking about are specific biomolecules. They are specific to living beings. I've shown you here 3 examples: DNA or RNA, the carrier of the genetic information, proteins, the so-called workhorses of the cell, and lipids, which make up the membranes of the cells. And I added a little thing here, water, which is not a specific biomolecule, but because all living things rely on water and water is everywhere, it's also something that we typically have to consider. As I said, everything is made from molecules, and as Richard Feynman said, everything that living things do can be understood in terms of the jiggling and wiggling of atoms. That's basically the approach that we are taking here, the bottom-up approach to understanding life. Luckily, there is a way to get the wiggling and jiggling of atoms, which is essentially the trajectory, the positions of all these atoms as a function of time. We have a method that can give us that. This method is called molecular dynamics simulations. This is one of the primary tools that we're using in my computational lab. And this is based on a very simple idea. It is based on Newton's second law, F equals m times a. So what this says is, if we know the forces on all the particles, then we can equate these to the accelerations, which are the second derivatives of the positions. And this is simply a system of coupled differential equations. The interactions between the particles, as shown here with this water molecule and these 2 ions that interact through Coulomb's law, we can simply derive from experimental data. Solving these differential equations, however, is pretty complicated, so we use some of the biggest supercomputers in the country to do this. Now, what do we do when we have this trajectory? Well, you can see that this is actually pretty complicated. You have thousands and thousands of these particles moving around. You have to somehow make sense of this wiggling and jiggling. And here we are using statistical mechanics to extract observables from these trajectories. So for instance, the second equation shows the radial distribution function, a way to characterize in a rigorous manner the environment that a particle is in. And we also use other approaches like Bayesian approaches to derive other quantities from our trajectory. Now let you tell me what we are doing in my lab. How are we using molecular dynamics simulations? I'm showing you here a textbook picture of a cell. And you notice that the cell is enveloped by the cell membrane. So this is the barrier between the inside and the outside. Now, the question is, how does the cell actually get nutrients into the cell or gets rid of waste products? All of this has to go through the cell membrane, which is really like an impermeable barrier. And this transport process across the cell membrane is really important in many parts of the body. So for instance, it's important in neurotransmission, in nutrient uptake in the gut, but also to control the working of blood cells. So for instance, the pH control in blood cells relies on these transport processes. But how does the cell manage to move molecules across the cell membrane? The cell membrane is made from lipids. It's like sort of a plastic bag around the cell. It's basically impermeable. So what you need inside this membrane is some sort of gatekeepers, some doors, or in this case, membrane proteins that span the membrane and that open and close. Here is a membrane protein that we are working with. It is a so-called active transporter protein, and you can see here 2 structures that we got from our experimental collaborators, which they solved with the method of X-ray crystallography. We have one structure in the so-called up conformation, in the upper left and one in the down conformation in the lower left. These two conformations are a critical part of the so-called transport cycle of this protein, because what this protein does, it cycles between this up conformation and the down conformation. And this is part of the transport cycle where the protein operates out of equilibrium to move sodium ions from inside the cell to the outside in exchange for a proton that moves to the inside. And this is one of these proteins that regulates the pH in blood cells, for instance. So one key question is how this conformational change happens. And this is something where experiments have a hard time giving us detailed information, so we use MD simulations. Here I'm showing you a movie that is actually one of these simulations where I removed the membrane and the water so that you could focus on the left-hand side. And as you can see, this blue part of the protein moves downward in this sort of very characteristic two-step process. And this is the first time that we can actually visualize what this conformational change looks like. No one else has seen this kind of behavior before. And using the information from these simulations and others, we can start piecing this transport cycle together and using a kinetic model derive the overall speed of this process. So what this approach allows us is to go from the structure that we got from experiments to a prediction of the overall function of the protein. So I hope I could give you an idea of what we're doing in my lab. So if you want to know more, there is the website that you can go to, and please feel free to contact me at my email address, oliver.beckstein@asu.edu. I'd be happy to answer all your questions, and I'd be happy to see you here at ASU. Thank you.